Nice. All right, perfect. Got the windows all set up. Okay, so this will be an overview presentation, mostly covering motivation and like our current approach. Basically, we've been working on this fMRI foundation model project off and on, as Paul said, for a while. Most recently, we like started it back up in June, and it's been mostly just us at SoFon working on it. And now we're opening it up for everyone to get involved. So I'm just going to be trying to catch everybody up with where things are at right now. Okay, so some background on what an fMRI foundation model is even. So it's pretty simple. The idea is you take this raw, noisy, complicated fMRI data, which is structured like a 4D time series of 3D volumes, and we're passing it through a big over-parameterized neural network, and the goal is to get out these neuro embeddings. So basically just like high-dimensional embedding vectors that capture important information that can distinguish people along important clinical dimensions. Like, for example, you might have clusters or subspaces that emerge in the embedding dimension that differentiates people with neurodegenerative disease from people without, or people with ADHD from people without, or depression from people without, or subtypes of these different conditions. Basically, the raw fMRI data is complicated and noisy, and you can't make sense of it directly, but we're going to pass it through a neural network and get out a structured embedding that we can then make sense of and use for downstream applications. Okay. So why do we want to do this? Why aren't we satisfied with our current approaches in translational neuroscience? So people have been trying to apply brain science in general to impacting people's mental health or impacting the status of treatment for neurodegenerative disease or neurodevelopment disease. It isn't really working, and this is like a widespread observation in the general field. So like this is some quote and figures from a recent book, which is pretty good, called Elusive Cures, from a prominent researcher in neuroscience, Nicole Rust. And she makes this figure in her book, which shows on the plot on the left, there's lots of papers being written every year on new, interesting neuroscience findings, but these are not really being translated into new treatments or new drugs to actually impact, people's brain and mental health. So like, why is this happening? What's wrong with the current approach in neuroscience, and why isn't it translating into actual clinical application? To take another perspective, this is a quote from the director from the NIMH, who was the director from 2002 to 2015, Thomas Insel. This quote is also taken from the same book. Basically, he's just saying that they funded a ton of stuff, they spent a ton of money on neuroscience, basic neuroscience, and it didn't really convert into important like clinical applications. And so if this isn't working, like obviously we should try some other different things. And people are going to be trying lots of different things as the field, looks at this issue and tries to make progress. Obviously, we don't have all the answers. We're not going to pretend like we know the answer to this big problem, but we can try some stuff. And the thing that we're trying is this foundation model strategy. And the gist of the foundation model strategy, I assume everybody has kind of heard the term foundation model, but the gist of it is pretty simple. You take a giant amount of data from some domain, you take a giant GPU compute cluster, and you merge them together to train a big neural network using some often self-supervised training strategy. And what you get out as a result is this, embedding machine, like I was talking about. And this sort of strategy has been applied in all sorts of domains. First, it was applied in classic machine learning, deep learning type domains. And then now more recently, it's been applied in scientific domains, like we're seeing here, 3 examples from the past 3 years, where the foundation model strategy was first applied to retinal images, to great scientific success, digital pathology images, which, as it turns out, we'll hear about tomorrow from Daniel in more detail. And then most recently in this year on like neural cell recording data. So in each of these cases, the strategy is working, where you take the data, large amount of data from some domain, combine it with the compute and some appropriate model and training strategy, and you get out something that's at least scientifically useful, and we hope will be ultimately like practically useful. In our case, that means like useful for clinical, mental health, brain health applications. And so this is the goal, and this is why we think it might work. So with that motivation on what we're doing and why we're doing it and why it might work, let's switch and talk about functional MRI data sort of more specifically and what the challenge of modeling it is. So I would guess like there's a handful of people here that know functional MRI data very well and don't need the review, the basic review, but some people probably aren't so familiar, so I'll just go over it anyway. So I always like showing videos of data. So this is a video of raw fMRI data, and you see three slices, like a slice down the middle of the brain on the left, a slice going sort of this way, and then a slice parallel to the horizontal plane. And if you can pay close attention, you can see tiny variation in the pixel values as the movie plays. And this tiny variation is the fMRI signal that we're interested in. And this signal is specifically measuring fluctuation in blood oxygen level. And the way the brain happens to work is when neurons fire, the body oversupplies oxygenated blood to those firing cells. And as a result, you see this increase in blood oxygen signal. And this is what fMRI measures. And the most important point is that the fMRI signal is a tiny fluctuation in the relative magnitude of the overall data. So it's only 1 to 2% of the overall variation that you can see. Okay, so how do we go about modeling these data? So like I said, it has some like critical challenges that we should be aware of and like key features that we should keep in mind when we're trying to think about how to model fMRI data. So first of all, the signal to noise ratio is very low. Like I said, the fluctuations are tiny, 1 to 2% changes. Most of the contrast that you see is actually due to differences in tissue type and not really related to the actual neural blood oxygen signal that we're interested in. The signal is very spatiotemporally smooth because the vascular system is kind of smooth, and brain in general, nearby areas are correlated in their activity. And then probably sort of the most subtle issue that people coming from outside fMRI don't often appreciate is that the actual signal that we're interested in, the blood oxygen signal, is concentrated in the thin ribbon of cortex. So like you can see in this figure on the left, bottom left, the cortex highlighted in white. Like this thin ribbon is where all the neuron cell bodies are, and this is where all the blood is being supplied and where all the bold signals being measured. And so it's really only in this area, as well as subcortical structures, which are not shown, where the key data that we're trying to model lives. The rest of it in the interior of the brain and definitely outside of the brain is not really of interest. So that's one key feature. And then finally, another important feature is like the brain has a standard map where roughly the same, each person has roughly the same areas and roughly the same positions that do roughly the same things, like modulo some wiggling around. Mostly the brain has a consistent map, sort of similar to how like the Earth has a consistent map of continents always in the same place. Like the brain has a consistent functional and anatomical map. So we should really leverage these properties when we go about trying to model the data. Okay, so with that sort of context in place, we can talk a little bit about what the current approaches are that have been tried in the past couple of years for modeling fMRI data. So one of probably the most common approach is what's called a parcellation-based approach. So this leverages these standard brain parcellations, which have been developed in the neuroimaging community over the past 10 or so years. And these basically carve the brain into these tiny areas, like I was describing. And these approaches extract the time series for each area by averaging over the many measurements within an area. And then they model this parcellated time series, which has reduced over the number of dimensions within each of these little parcels, it's called. So I can just go into this a little bit more detail, I guess. Like each one of these parcels will generally have about 100 tiny cubes of fMRI measurements in them. And so these parcellation approaches, they average over those 100 cubes to produce just one number for each fMRI time point. And so as a result, you get this parcellated time series matrix, which is number of ROIs going down the rows by number of time steps going across the columns. And this is a matrix around size like 400 by 200 time points, for example. And sort of the key weakness with this is that it loses a lot of information. So whereas the fMRI, raw fMRI data is like a big volume of 100 by 100 by 100 tiny voxel cubes, this is now just 400 by number of time steps. So you've lost a lot of information doing this. By contrast, another approach at sort of the other extreme is a 4D volume-based approach, where instead of doing this parcellation averaging type strategy, you just take the raw 4D fMRI data directly. And this has no loss of information, but at the same time, it's quite complicated to model the data directly. You're not doing any of the you're not taking advantage of any of the known properties of the cortical folding or the map of areas or any of that stuff. It's just a very raw native approach when you try to model the 4D fMRI data directly. So we can kind of put these two classes of approaches along this in this space of sort of more structured versus less structured approaches. And something we've all probably learned over the past however many years is this lesson called the bitter lesson from Richard Sutton. And basically it says that ultimately the approaches to AI machine learning that win are those that take advantage of compute and data, and whereas like clever, sort of cute and highly structured approaches that have nice ideas often don't win in the long run. So in this plot, you have these multiple curves, like a very structured curve in blue, which has lots of great ideas and plateaus early as you add compute and data, and then a very unstructured curve in red, which doesn't work initially, but once you have enough data and compute it, it works well. And we can kind of think of these two approaches that have currently been tried as the parcellation one is the blue, highly structured curve, and the volume voxel-based one is the red, unstructured curve. And kind of the motivation for our current strategy is to basically ask what would be a good intermediate between the parcellation approach and the highly structured parcellation approach and the unstructured volume-based approach. Because currently we don't have enough compute and enough data to support really the extreme unstructured strategy, in my opinion, or at least we should have an intermediate approach that is between these that we can try out. So this is the main sort of driving question motivating our current idea. And so the answer that I'm proposing or that we've been working on is basically flat maps. So we've been working with these flat maps for a while. And what this is you take I can show you the video. You take the brain, you reconstruct the surface, you inflate it, and then you make some cuts so that you can then flatten it out. And that was maybe fast. Just play it again. Yeah, so the key point of this is that you're taking the voxels, which are scrunched together in a complicated way, and you're flattening them out so that now sort of neighborhoods in the flat map space correspond to neighborhoods along the cortical folding. And this way you get to have a nice property where the relevant spatial relationships are captured directly in the data, as opposed to the model having to basically learn to figure out which parts of the brain are connected itself. Okay, so if that didn't quite make sense, the main takeaway is that flat maps as a representation of the fMRI data preserve more information than the parcellation strategy, which reduces all the local areas down to one number. And at the same time, it's more structured than the 4D volume strategy, where there's less stuff that the model has to learn from scratch and more information sort of given to it in the form of inductive bias. Okay, so this is the sort of the core crux of the current idea. And to try to drill into this a bit more, this is how the pipeline works for generating these flat map images. So you take the raw volume fMRI here. You can reconstruct the surface using separately collected structural MRI. And so that's shown here, where the cortical surface is fit to the gray matter ribbon in the structural MRI. And then you can combine these two to map the fMRI onto the surface, the cortical surface, and then do the flattening that I just showed in the previous video to get the final flat mapped fMRI. And so this process is sort of a computationally expensive pre-processing step that thankfully is done for us and has been worked out over the past 10 years or so of neuroimaging community development. And so we're just leveraging this in order to be able to project our data down into a representation that has the right structure that we would like. Okay, so the main takeaway that I want to emphasize here is that this flat map representation is a representation of the fMRI, and it is an image, like a flat image like this. And the reason this is important is that once we have our data represented in image format, the key thing that we gain is the ability to pass them through standard vision models like vision transformers. And so to spell this out more, standard vision transformers, they just need a 2D image, and they patch the image into the sequence of square patches, and then patches are passed through the vision transformer directly. And because we've rendered the fMRI data down into a regular 2D flat map image, we can now just use vanilla models like vision transformers directly. And this is a key advantage of the current strategy. Our current approach, so really any approach could work with the flat map rendered fMRI data. You could try any vision self-supervised learning method that has been developed in the broader computer vision community here. The strategy that we've been working with currently is the mass autoencoder framework. And so this is work developed by Kaiming He in 2022 from Facebook Research. Probably most people are familiar with mass autoencoders. But just to sort of review, the idea is this is a self-supervised learning framework that basically trains vision transformers to reconstruct these unobserved mass patches given a sparse set of observed patches. So you have an input which is mostly masked on the left. The unmasked observed patches are passed through an encoder model, which then computes embeddings for the observed patches, and these pass through a smaller decoder, which makes predictions for the unobserved mass patches, which are then compared to the original target patches from the data. And so this is a nice self-supervised learning framework compared to some other alternatives because it's quite simple. The objective is well-grounded. You can interpret the reconstructions nicely. The loss curves are stable and don't suffer from any non-stationary dynamics like you see in some other like student-teacher type methods. And then sort of most importantly, something that the authors of the mass autoencoder paper show is that it doesn't require data augmentation. And data augmentation is kind of tricky with fMRI, so it's nice to start with a framework that doesn't rely on it. So this is our current approach, and we have some initial results on how this model is doing to show. So just quickly to get a sense of the qualitative performance. So this is a figure showing the masked reconstructions, where on the right you have the raw target flat map fMRI data. On the left, far left column, you have the masked input that the model sees, where the observed patches are, the values are shown, and all the unobserved patches are just have white background. And then in the middle, you have the prediction from the masked autoencoder model. And as you're going down the rows, you have increasing time steps. So this is a plot for one example fMRI sequence, and the masked autoencoder is making predictions for each time step. And the main thing to notice is that the model is able to capture sort of the precise signal pattern that changes over time without being able to reconstruct any of the noise that is present in the target image. So like most of the high frequency signal that you see in the right-hand side column is going to be noise and unpredictable. And the model is able to reconstruct sort of the signal and sort of implicitly denoise the data because it's unable to predict like unstructured, unpredictable noise. So this is kind of the qualitative visualization of that the model is making sensible predictions and reconstructing the data well. And then for a more quantitative look at this masked reconstruction performance, we can see this plot which shows very nice so-called scaling laws. So as you increase the size of the data, it's been shown quite famously now that, for example, in language modeling, that as you increase the size of the data, the performance at predicting the next token increases with sort of a log linear relationship. So this figure on the right from Kaplan is showing dataset size and tokens on the x-axis in log scale, and the log loss, next token prediction loss, on the y-axis, and it goes down as a log linear function of dataset size. And like quite interestingly, we see the exact same pattern here with the masked autoencoder loss. Like the scaling trend is so good, the fit is so good. I would think it's maybe a fluke that it came out so well this time, but at the very least, like it's an impressive, very impressive fit of the power scaling law trend that you see in language modeling and vision modeling and so forth. So we see as you increase the dataset size from 500,000 frames up to over 7 million frames of fMRI data, the masked autoencoder loss is going down like a log linear function. So this is very nice to see. This is something we've been hoping to see in modeling fMRI data for some time. It's kind of a prerequisite for believing that foundation model strategy can work, to see that the models respond to increasing data, and that as you increase the dataset size, the models are able to predict more and learn more. Okay. So this is just masked reconstruction performance on held out data from the training data set. And then this is interesting and a nice, sign and sort of sanity check, but it doesn't capture the full story. What really matters is ultimately the downstream performance of the features that you learn. You want the features that you learn to be useful for tasks you actually care about. Mass reconstruction is not in itself a task that is, important for application. So in terms of downstream evaluation, I would say that we have some quite promising early results. So this table is showing downstream classification accuracy on 3 tasks, which is basically like cognitive state decoding in the top row, where you're trying to predict which sort of cognitive task a person is doing in the scanner out of 21 possible different tasks. In the middle row, you have a visual decoding type benchmark, like similar to the image reconstruction benchmarks that we've done in MindEye and that have been done in the literature quite often, although this one is a simpler just classification benchmark. And then on the bottom row, you have sort of a standard benchmark evaluation, which is the sex classification for UK Biobank. And I'll just kind of distill this down rather than go through each column to say that we're getting really good performance on the ACP cognitive state decoding, where the current state of the art, which has been reported, is around 94-ish%, and we're getting over 98% and basically approaching ceiling on this task. So we're already sort of convincingly beating the state of the art in this particular task. On the bottom row of sex classification, we're kind of in the vicinity of the current state of the art. So like previous methods get between like 84 and 88% on this task, and we're kind of around 84, 85-ish. So we're not hitting the state of the art level yet, but it's kind of got a sign of life to it, where if we continue to work with this framework, confident we can get some improved performance. Like, we can get into the details more later, but there are quite a few differences between what we're doing here and what the other authors have done. And then finally, the visual decoding task doesn't really have a close comparison to discuss, but I still think the results here are super interesting. So this is basically showing something I didn't describe yet, is just this is showing basically 0-shot visual decoding performance on a held out subject. So the model is trained for this shallow linear probe style evaluation on 7 out of 8 subjects for the natural scenes data set, and then it's being tested on a totally held out subject, and still it's able to decode the performance. I mean, it's able to decode the visual category of the scene image from the fMRI data of this totally unseen subject with quite good accuracy, almost as good as what it achieves on the in distribution subjects that it was trained on. So it gets around 21% accuracy on the new subject versus 25% on the training data subjects. And this is really good, but close gap between the in distribution subjects and the new subject, which hasn't really been seen before in visual fMRI reconstruction tasks. Okay, that said though, like, this is still early work. We've only been working on this for around 2 months or so since we restarted, and there's still a lot of stuff to do. So one obvious thing that we need to do is basically train on more data. So right now we're only training on data from the Human Connectome project, which has around 2,000 hours of fMRI data in it. And we have recently gotten access to the UK Biobank, which has about 5 times as much data. And so we just need to add this to our training data set and see if we can improve performance. There are some tricks to work out because we've done some initial experiments with this and noticed some instability in training. And so there will be some things to figure out here, but I think this is an obvious direction to improve what we've done so far. Okay, the next thing we need to do is basically just have better direct comparison to previous works. So I was describing the comparison between our current results on the UK Biobank sex classification versus some of these prior works like BrainLM and BrainJEPA. And the numbers, if you compare them directly, are sort of in the vicinity. We're not doing quite as well, but we're beating some of the earlier methods. But you have to take this with a critical grain of salt, because these comparisons, you cannot compare the numbers directly. There are tons of differences between how we're evaluating the model and how these previous authors evaluated their model. And so we really need to implement a direct comparison to these prior works in order to know how our model compares. So this is something we need to do in the next couple of weeks. And then the last thing that the last immediate thing that we need to do is basically a bunch of ablation experiments to figure out what is the best way to train masked autoencoders on fMRI data. So we're adopting this framework from the literature, this masked autoencoding framework from the literature. And the original authors of MAEs did all these ablations of masking ratio and data augmentation and how exactly to formulate the loss and all sorts of things to figure out what configuration works best for natural images and video. And we haven't done any of this for fMRI, and it's no reason to think that the settings from natural images and video will transfer directly to fMRI. So we need to do this in order to figure out basically how to optimize our model for our particular data. And so to just summarize that into a short thing, like we need to make this table. We need to make a version of this table to include in our sort of preprint paper that we're working on. Okay. So these are the main steps that we have next to do, critical key next steps. But there's also a ton of other things to work on. So I want to do some qualitative analysis of the model. There's lots of interesting qualitative analysis that we could do. We could look at attention maps. We could look at sort of PCA visualizations of the feature maps. We can visualize the embedding space, like I motivated on the first slide. We want to implement some more evaluations, some more clinical angled evaluations, obviously. And then we also want to try to do some effort to standardizing benchmarks, or at least sort of help the field move towards more standardized benchmarks to address this issue of incomparability across works. And then there's a bunch of other things we want to do as well. I'm not going to go into each one of these bullets, but just there's enough stuff to work on. We're going to be working on this for a while. There's plenty to do. So that's all I have for the overview presentation. I know this was probably quick. For people that don't have much background, there probably wasn't enough background. For people that have, who know all this stuff, maybe it was too slow. But I mean, this was meant to be just a quick overview, and we can have discussion now, and we can also talk more during, over Discord and have follow-up conversation. But yeah, so I'll pause now, and then, yeah, people have questions we can discuss.
Have you turned the entire UK biobank into flat maps already?
Yeah, great question. No, Paul just finished downloading them like yesterday or today. I think he sent the message today. So we've converted a small fraction of them to flat maps to use for the evaluation, but we haven't converted all of the data to flat maps. So we're going to do that like next
And
week.
have you got around the all the legal issues of, like, last we talked about you had to use the UK biobank in their specific cluster, right?
Yeah, after nearly half a year, maybe a little more actually, of going back and forth with UK Biobank, we finally got the permission to download the data outside of their crappy RAP system, research access portal system, which is very important because we wouldn't be able to train the models within their specific cluster. So finally, yeah, we were given the ability to download it, and then finally finish downloading it to our own means, and then have to do some conversions. But yeah, it's good to go now.
Great. So they finally saw reason. Then I assume the flat map conversion will go fast enough with your cluster.
Yeah, that part will be fast. Maybe take it a day, but then we'll have all the data. and it'll be like 5 times as much as we have.
Are you guys looking to use any other data apart from UK Biobank?
Yeah,
I have ADNI, which is pretty much pre-processed, but um, if you want to use that, we can just convert it into flat maps.
yeah, I think ADNI would be great. We've definitely wanted to add ADNI. I think there's some data sets that are more suitable for like pre-training and some that are better for evaluation. I feel like I don't remember the size of ADNI, but I feel like it would be better for evaluation. I know I've seen
I
lots of or a few. Yeah,
I don't know how much is it on frames, but the total is around 1.54 terabytes. But that also includes like structural data, so I'm not really sure
yeah, and that
about.
might be the raw data. So yeah, we can look at it. We for sure will want it. I've seen evaluations that use ADNI, and yeah, basically any data that we can get into the flat map format, we might as well add either for pre-training or evaluation. The main data set that I want to add is ABCD. That's like the last
Mhm.
of the big sort of 3 data sets for fMRI, HCP, UK Biobank, ABCD. ABCD has been quite hard to get hands on, and they're sort of pretty restrictive with the data access. And even if you have access, it's kind of tricky to download. System is often down. So yeah, if anybody has a line on ABCD data, that would be nice.
I think there's multiple centers working with ABCD. Like I interned at a lab at Stanford that I think used to work with it. So I don't know if we can like pull on old strings, but try.
Yeah. I've technically worked with ABC data, so we've processed it and used it in our labs. I mean, in my previous labs, I can try to see if there's a way to get the data for just the flat maps.
That sounds good.
Is
Are you-
ADNI the Argonauts data set, Argonauts 25 data set? Because I think it would be interesting to combine the video data with the image data from the NSD image data, basically.
ADNI is a separate data set which essentially consists of people coming in for MRI scans over long periods of time. Like there are some subjects who have taken scans in 2018 and they're undergoing scans in 2025, specifically for Alzheimer's study. So it's mostly longitudinal scans that you can track off with ADNI. Yeah.
Yeah, but the Argonauts. Yeah, sorry. Go ahead.
Yeah, I just wanted to mention that I think whatever the data that we're working with fMRI foundation model is all resting state. And not sure how it's gonna work with if it's like task-based, like CNeuromod, the same thing that Argonauts were using.
Yeah. So the HCP has a lot of resting state. It's definitely mostly resting state, but it also has quite a lot of task fMRI data. There's even some like movie watching data for a small number of like a small fraction of subjects. So there is some task and other data in there. Although, yeah, the majority is resting state.
And is it generalizing well over the task-based ones?
Yeah, I mean, so on the task state prediction, which is clearly like a task-based evaluation on HCP, although held out subjects, performs super well, like almost 99% accuracy. So it's
Okay.
able to do that task, no problem. And then on the more interesting, like much more difficult NSD sort of visual classification task, it's doing well. We don't have a comparison because nobody has really done this task before, but it's doing much better than baseline methods, which themselves are quite strong. Like the patch embedding, I didn't go into this, but our baseline method, which is what we call patch embedding baseline, gets basically matches the previous state of the art on human connectome state decoding, gets around 94%. And then on the much harder NSD visual decoding, our method does twice as well as that one. So yeah, it's definitely able to get something out of the task-based data, even though it's only trained mostly on resting state.
Cool.
Did anybody else want to? Was anything else unclear? I think maybe Claire, you've never seen any of this stuff before. Did you have any questions? Were any parts confusing?
No, honestly, it was pretty like I know more of the imaging piece of it than the immediate approach, but no, I think it was pretty straightforward. I think diving into exactly what you did and when that would be useful, I think, for me. But
Okay, cool. Okay,
yeah,
cool. I'm glad it was mostly clear. We can then actually do some of this stuff of diving into stuff that we actually did by looking at the code a little bit. This was one of the other things I wanted to do with our time. We have like 30 minutes, so we could spend some pretty casual time looking through the code and then yeah, testing it out. Okay. So this is the repo, fmri-fm repo at medArc. So the repo is not currently public. It should be public soon. We'll decide exactly when to do that. But like the plan was to start the project, get the initial like preprint done. We have a preprint we've been working on to write up our current results for a workshop at NeurIPS this year. And then we were going to make the code public. In the meantime, I'm happy to add anybody that would like access to the code directly to the repo. So I've been doing this already, and I think some people messaged me today to add them to the repo. I haven't done that yet, but I will. And then if anybody else wants access, I'll add you. And then hopefully before too long, everyone, it will just be public, so we won't have to do that anymore. So this is the repo. The README is hopefully pretty straightforward. There's a link to some background, which is kind of just the story I just gave in the talks written up. And there are some links to relevant papers there. There's a reading list with lots of relevant papers that you could go through. I'm sort of covering, closely related fMRI foundation model work, as well as important like work in brain behavior prediction, sort of with more classic shallow type approaches. And then like lots of background on visual self-supervised learning, where we got a lot of inspiration from. So we can go through these papers, or you can go through these papers and basically get up to speed on what the literature for the project is. The installation of the project should hopefully be super easy. I think I will try to demo this now by installing it from scratch. Start it. Good. Just share my whole screen. Okay. Okay, so I'll just clone the project. And then we use UV for just like setting up the environments, managing dependencies and stuff. Like I'm sure everybody, or like a lot of people, are using UV. If you're not, definitely should. It's much better than whatever I was using before. So now that I've cloned the repo, you can just do UV sync, which will install exactly the environment that is used for all the experiments. So there's no clashes in versions or any stuff like that. And that's all there is to installing the project. Okay. So a little bit of going into the code base structure. So we're trying to basically keep the code base structure simple and understandable. And one way to do that is to avoid having like a deeply nested structure with all kinds of abstractions and complicated interfaces and having to jump through a ton of files to figure out what anything does. And so we just have a pretty flat structure that has just a few main important files to look at. And in general, the code is forked from the original MAE repository. So most of the files are just versions of the modules that they had. So there's a main pre-training script for training the MAE on HCP, and then there's two downstream evaluation scripts for classification and regression tasks, downstream tasks. I've since merged these into just a single downstream evaluation script, and I'll push that change before too long. So these are the main entry point scripts, which would be good to read and just give you a pointer into the entire code base, more or less. And then there's some model implementation modules, the most important, which is obviously the models MAE. So this is basically a copy of the original MAE model implementation from the original code base with a few small changes. Basically, the only important change that was needed to be made to the code base is the introduction of these masks. So the only difference between the fMRI flat maps and natural images is the presence of this background of all 0 unimportant values. And so the only change we really had to make to the MAE is to account for these, invalid all 0 background values. So we did that by integrating this image mask argument, which is just a mask of the valid pixels, and then update all the logic of the mask autoencoder to account for that mask. And there's really no other difference in the model implementation for the MAE. Yeah. So the main pre-training script is like, I don't know, I don't really enjoy reading these. It's just there's a ton of boilerplate, which is all the same stuff you've probably read many times of creating the model, reading the config, loading the data set, running the training loop, running the evaluation, saving the stuff. So we can go through that. I mean, you can go through this later, but it's pretty straightforward, and it's just ultimately a copy from the original MAE codebase. I would say the only part of the codebase that has the most unique stuff, the most changes, is this flat data module. And this flat data is just where all the flat map specific stuff is defined. So like the utilities for creating the flat map dataset, we're currently using web dataset format for the storing the flat maps. And so there's utilities for creating that. There's utilities for creating this like clip dataset, which is just like a standard get item style torch dataset. There's transforms and utilities for extracting random clips and all sorts of stuff that we need. Some utilities for masking. So this is kind of where all the main difference between our approach or our current work and MAE is just changing the data. So the only place where there's interesting stuff happening in the code base is mostly data related stuff. And so this flat data module is kind of where most of the changes are. Okay. So what else did I want to discuss? I think for the rest of the code base, it would be good to maybe look through on your own. I'll tell you in advance that like some things that are not currently documented as far as how to do are probably the things that you want to do first, which is actually training models. So the data sets are not sort of released yet, so it's not possible to run any training, but we'll be getting the data sets released pretty soon, probably same time as publishing or making the code base public. And so that's stuff related to running training and where to get the data and stuff like that is currently missing from the documentation. But will be added soon. In the meantime, what we can do is run this little quick start notebook that I made. So this will just give you a sense of, what the model does, proof that the model runs, stuff like that. So I have this version of the code base that I just cloned, the clean version that I just cloned. I've got this branch that I just started, which has the quick start notebook in it. I'll merge this branch before too long. And we can just run through the notebook. So I uploaded some example data and an example checkpoint to Google Drive, and I made this public. So should be able to download this stuff. It just has like one shard of the ACP flat dataset and a checkpoint for a pre-trained VIT small. And if you want to follow this stuff along, it's in the quick start branch. So you navigate to the quick start branch, notebooks, quick start notebook. And yeah. So you can get this notebook and run it and get the data from Google Drive. Yeah. So we'll just run this notebook on our machine. And I'm just running this on my laptop, so you don't need any GPUs or anything to just run this notebook. So we've got some importing the models module, importing some stuff for creating the data set and transforming the data, then importing a utility for plotting predictions. Just all we're going to do in this notebook is just running the model, plotting predictions. Okay. I think I have to, oh no. I think I will actually have to download the stuff. That's not good. I deleted the version before. All right, we can see how long the download takes.
I guess what would be the most immediate thing people can help with, Connor?
Yeah, that's a good question. Yeah, I think the most immediate thing is to try to, so like I said, the three main tasks we have are to train on the UK Biobank, to get better comparisons, and then to run the ablations. I think the first and the last one are pretty like compute heavy tasks, but the getting the comparison stuff is something that doesn't require very much GPU access to get started. So I would say we should do, I think people should be able to contribute to that. So that will specifically involve getting so what we want to do there is to get these code bases for these comparison works, BrainLM, BrainJEPA, maybe a couple others, but mostly those two to start. Get the code bases for those works, integrate them into our repository, get them running, so which will require some small modifications to get them running within our environment, and then figure out how to like load their checkpoint and then run our evaluations using their loaded checkpoints. And so this will mostly involve, some coding and wrangling the dependencies and a bunch of kind of annoying troubleshooting that doesn't require a lot of GPUs to start. And so we've already done a little bit of this, like Daniel was working on this a little bit, but we still need to, finish it up and get it working and getting the evaluations running. So I think that would be a good area to work on. I already asked Mahir separately if you want to work on this. I don't know if you do, but if you wanted to work on it, I think that would be great. I'd really appreciate having you work on that stuff. But yeah, I think getting these comparisons running would be really useful.
Okay. And then for the other two things you were talking about, we can have people help with the non-evaluations of Brain Shepherd, Brain LM as well. If we can add them to like our Lightning compute and then have the data already downloaded. So if you're specifically interested in that, also let us know.
Yeah, I think for that, there are a bunch of issues that I've opened. I'll be opening more issues, but you can always look through the issues and see what's available to be worked on. And any of these open things are good to work on. And basically any of them that you feel like you can make traction on are good to go. And generally, most of them will involve two things. They'll all involve some implementation, changes to the code base, and then also some running of experiments. And I think like if you can do the implementation part, then when it comes time to test it and run experiments, by that point, we can have added you to the compute allocation, and you can be able to run the experiments on your side, or somebody that has the compute already can run it on their side. But like the implementation side of any of these open issues, as well as issues that I'll be opening later, can all be done without any compute access. So like there will be all of these implementation changes we need to make for running the ablations. Any of those code base changes can be done without being on the compute setup.
So I guess, like practically speaking, the workflow is take a look through the code base, run your quick start so you can get a little familiar with it. Look at the open issues that are that you wrote up here for sort of ideas of directions to go, or just look through the code base yourself and see if you can improve it in some ways, and just message in the Discord channel about the directions that you're exploring or the problems that you want to work on, and we can figure out a best way to make us the collaboration work for that. And also make a fork, right? So you make a fork of the repo and
yeah, that's right. Yeah, so to get started working with the code base, you just make your own fork. Some people already have. Daniel, Daniel's got a good fork going. Yeah, so you create a fork and then make your changes in the fork and then open a pull request back to the main repo. Some of this workflow is documented in the contributing guide, and so you can go through the contributing guide for some more description of this. But yeah, basically fork, open a pull request, draft pull request to start, saying I'm working on this issue. Here's my work in progress stuff. And then once it's ready to go, convert the pull request into ready to review, and then can merge it back to the main code base. Yeah. Okay. Yeah, these were good reminders. Can you think of anything else that we should talk about, Paul?
We're going to be meeting every week at this time, and but these weekly meetings, aside from today is like an introductory sort of get people to know what you've accomplished thus far and sort of like warm people through general protocol. But usually we do these weekly meetings as a sort of like catch people up on what people have done since the last meeting, sort of summarizing that what people have done and allocating tasks to people who want to help out. And then we are making progress towards, get retraining to work on bigger data, evaluations to work across a couple different foundation models so that we can show that we have great results, and then we'll write it up together and publish it somewhere.
Yep. Sayed, I think you had your hand up.
Maybe joined like quite like 10-15 minutes late, but I just wanted to know if you have already shared the timeline for this project, maybe.
Yeah, that's a good question. I think it's not so firm what the timeline is. Yeah, I don't know if I've really ironed out what the timeline is exactly, but I think we're looking certainly to submit a paper within somewhere between 1 month and like probably 3 months, probably roughly something like that. So we currently have like an initial workshop paper written up, which is like a short 5-page paper, mostly going over what I've already talked to you guys about. And then, yeah, we want to expand this, adding the things that I talked about, and write up a full paper for a conference. There's iClear at the end of September. That's like coming up very soon. So it would be difficult to have very many new contributions in that time. But then after that, there's quite a few conferences after that. So somewhere in the next few months, we'll be looking to finish the main tasks like the main 3 things that we were talking about, and then write up paper for submission. Yeah. Cool. Yeah, was there another question?
Yeah, so sorry to interrupt. I have a question. So when you work with like masked autoencoders, what is your vision into like making it better for this specific case? Like one idea which I have in mind is maybe to work with loss functions of masked autoencoder, because right now, as I understand, it is like very simple. It just reconstructs the average pixel intensity of the patch which is masked, right? So in this case, you are what I see here is that masked autoencoder is basically overfitting the objective to not to learn useful visual representations in global terms, but like to learn basically to reconstruct just like everything on a pixel value. Is there any way to develop this further, in your opinion? For example, like the foundational algorithm of representation learning here.
Yeah, I think that's a pretty deep question. I think so. A couple things to say. So although the mass autoencoder objective seems quite low level, where you're just reconstructing pixel values, the previous work showed that it nonetheless results in model learning, like useful, rich representations shown by good downstream performance on so for example, if you train a mass autoencoder on ImageNet, you're just reconstructing pixel values, but you evaluate on ImageNet classification using the frozen representations, and it performs well. So it just shows that even though you're training on this low-level pixel reconstruction objective, it still nonetheless learns useful representations. And you can understand why this would be, because you've structured the task crucially by using a high masking ratio so that the model can't just solve the task by a easy shortcut strategy, like by copying pixel values from neighbors or interpolating over spatial temporal neighbors. So by making the task hard with a high masking ratio, you force the model to learn basically the high level complex dependencies in the data, and you don't let it just predict the mean pixel value, that sort of thing. That said, I think there still is a lot to think about, especially in terms of how to refine the MAE task to make it an even better fit for the fMRI data. Like something that's unique about the fMRI data is that it is quite spatiotemporally smooth, and you can see this in the quality of the reconstructions being very smooth. And so maybe we need to do something to specifically handle this in a way that the original MAE didn't need to do. So there's lots of things that we could do along this direction, and we've talked about them in our own meetings quite a lot, and we could talk about them more later. I'm pretty interested in this direction. And so short answer, yeah, for sure, there are things to do to specialize the MAE task, even though it is shown to work even better for fMRI data. Was there anyone else that had a question?
Hello, I have a question.
Yeah.
Regarding the initial vision model that was pre-trained on the data, I'd like to know whether the team is considering making architecture modifications to the existing vision transfer model, or if there are also the plans to establish additional benchmarks using alternative models.
Okay, so was the question whether we plan on making architecture changes to the original vanilla VIT?
Yes.
Yeah, so related to that, one thing we've talked about are just adding on sort of the bag of free improvements that have developed over the past couple of years. I think Daniel knows these more closely, but things like swapping out the layer norm for RMS norm, swapping out the basic activation for the gosh, I forget what the other activation, basically swapping out the activation function, all these sort of things.
FFM.
Yeah, exactly. Yeah, thanks, Dana. I was blanking on the names. But yeah, all these sort of bag of free improvements we could do to the vision transformer. As far as trying to come up with novel improvements to the vision transformer, I think we can think about this, but we should also be careful because it's quite a nice feature, I think, to say here we've just been able to reuse what's already been developed for images and video onto fMRI without having to change it wildly. Whereas the other works, because they don't use a representation that is even amenable to the classic models, they can't do that strategy. So it might be not, certainly we should start with the vanilla architecture and carefully make changes and validate that they actually do improve things reliably. And because we don't have to make changes to the architecture to get it to work. Okay, Sam, you had a question?
Yeah, so very nice work. I was curious about, because you were mainly talking about sort of the spatial dimension initially when it comes to your choices for the approach, and I was curious about this temporal aspect. So you included some comparison between different patch sizes, and as you know, I was looking at Brain Japa the last few days, and they, for example, have these really long patches. Actually, I think it's 16 or so, and at a TR of 2 seconds, you have a half a minute basically, and so it was quite nice, but also surprising that performance basically improved the smaller your temporal patch becomes, if I got that right.
Yeah.
Is that were you expecting that, sort of, given the the bolt function? Yeah, I was just wondering what your thoughts were there.
Yeah, that's quite a good question. Yeah, paying very close attention, even though I didn't go into that part of the figure. Yeah, so two things to say on this quickly. When I went over the MAE architecture, I talked about it just from the perspective of the vanilla MAE, which is not spatial temporal. And I made this figure up just to be okay, we're given a single frame of the flat map and we're reconstructing the unobserved patches for a single frame. This is mostly just for convenience for explaining it, but really because fMRI have this temporal dimension, we're using the spatial temporal version of the masked autoencoder. So you don't just give it a single frame, you give it the sequence of frames. In our case, we give it 16 frames. This is the standard sequence length from the spatial temporal MAE on video. So we give it this spatial temporal sequence of 16 flat map frames, but otherwise the architecture is the same. And then, yeah, you pointed out this thing about the patch size. So the temporal patch size is crucial because it just changes the spatiotemporal granularity of the model, basically. If you have this input, which is 16 frames by height width of the flat map, and if you use a large temporal patch size, you're covering most of the temporal sequence with the entire spatiotemporal patch. You have pretty few patches overall, like small number of tokens overall, versus if you have a small patch size, small temporal patch size, you have many more tokens in the sequence. And yeah, I had this experiment, which I didn't talk about, where we vary the temporal patch size from numbers going down from temporal patch size of 16, which covers the entire frame depth, and which is the same as BrainJet, but they also use 16 frames. The TR is different. So our TR is 1 second and theirs is 2 seconds. So 16 frames here is 16 seconds compared to half a minute, but still 16 frames is a lot of frames. And we see, like you said, the performance improves with smaller temporal patch size. And this is kind of an expected, like known effect in sort of classic vision transformer work. People have noticed that smaller patch size often gives you better performance, even at the same image resolution. And this makes sense because you're just giving the model more token capacity to work with, more tokens to use as its working memory, more or less, and do attention over. And so you just basically have a speed accuracy trade-off, or like a compute accuracy trade-off, where smaller temporal patch size means more tokens and more compute and more accuracy, but slower runtime, basically. But it's definitely something we should think about more. It was an interesting result, and it's kind of one of the many ablations we need to do to figure out what the optimal temporal patch size is.
Yeah, cool. It seems like a pretty strong effect, especially on the
It
sex
is,
prediction
yeah.
there.
Yeah, it's quite interesting, strong there. I'm curious why the effect is so clear with the sex prediction. I mean, it's
So
pretty clear it's pretty clear in this one too. Especially if you look at these bars, which are the performance on the same, which is basically like the in-distribution subjects performance. It's the smallest temporal patch size doesn't work as well on the new subject by a small amount. But yeah, I'm curious why this effect on the sex classification is so strong. I'm not sure exactly why.
I was doing some experimentation with like a very different approach, so more towards the self-distillation, right? Like a much higher level objective. And there, I actually, I mean, I didn't do very comprehensive comparisons there, but I was seeing opposite trends actually. So I might want to try to get some sort of comparative results, such that I can better compare to what you're seeing. That might be interesting. Yeah, just to gain understanding as well.
Yeah, I think that would be cool. We should talk more. And we can also look at some of the stuff you're doing.
Sounds good.
Okay, so it's 9 o'clock. We wanted to be sure to end on time because there's more meetings coming up. I just wanted to thank everybody for coming by. Hope that the presentation made sense, and that I hope you'll all, if you're not already in the code base, give me your GitHub username, add you to the code base. The past couple of weeks, been working hard on the workshop submission, but like starting next week, I'll be getting back into the code base more and be able to help onboard people into the project and hopefully get sort of the next round of experiments and work going. So yeah, thanks everybody, and we'll be meeting next week and hope to see a lot of you then too.
Yeah, thanks so much, everyone. Great to see a lot of new and familiar faces. We'll keep in touch in the Discord. Connor can also share his slides. Yeah, thanks everyone.
Thank you, everyone.
Thanks for the presentation.
See ya.
Thank you.
Good.